So let me start. Actually, this is my first time to send out the video presentation. So if any problems may take place, my apologies in advance. Before starting anything else, my name is Yutaka Suzuki from the University of Tokyo. And I'm really honored to have this opportunity to join the meeting, first STOC meeting, which is, I believe, the cutting edge meeting of the spatial transcriptome, new technology, new platforms. So let me share my slide. And here, I hope you can see this slide. And today, I would like to talk about a progress report from the University of Tokyo, where I'm in charge of the sequencing collaborative. We ourselves are also working on the spatial transcriptome analysis, mainly for cancers, several species of cancers. So I want to share the information as much as possible with you, as soon as possible. So, and this is actually everybody is sharing the same enthusiasm about looking forward to the second generation, at the start of the second generation spatial transcriptome sequencing analysis. On one side, 10x Genomics has just announced the launch of a new platform, Xenium, where the subcellular analysis of the transcript analysis is firstly enabled by in situ hybridization analysis. They announced that 400 genes are the first targets for the hybridization, the making a probe, and the simultaneous analysis in the future will be possible with proteins by immunohistochemistry and SNV's detection in cancers. And the details is not disclosed yet, but they say they are making use of different technologies from Cartana and Read4, two venture companies the 10x Genomics purchased several years ago. And they have just opened a demonstration page and starting an ad for showing several examples. And no less important thing that no less important news that I was aware of was the BGI STOMICS serosig analysis. And they say there is maybe everybody is coming here to know the further details information about that. So I may not have to explain more details, but they say the resolution should be 500 nanometer, and they can also analyze, say, a quite big tissue, like the whole mount in situ of the mouse embryo, which is sometimes bigger than 1 centimeter, which is more than 10 times higher than the current vision platform. So maybe I'm taking too much time on the introduction, but let me continue. So, okay, I should have started from here, but my place, I'm in charge of a sequencing collaborative. This is my place, and there are three campuses in the University of Tokyo, and this is the latest campus, the newest campus, the most recently launched campus of the University of Tokyo, Kashiwa campus. Here, like this, perhaps the new things are, this campus is designed to start new things in biology, including genome science. So we are currently operating HiSeq are almost old-fashioned, and we are mainly shifting the sequencing focus to NovaSeq. We are possessing 3 NovaSeqs, and we have recently introduced T7-MZI, this one instrument, and also setting up G400, which is on request, by the way. So, and our place is being operated by about 15 technical staffs, and the mission of this place is to provide this sequencing as platform to the community. Half of the grant to support this place is coming from the Ministry of Education, and the other is coming from the Ministry of Health. So for the Ministry of Health part, we are mainly focusing on cancer sequencing. And at the same time, with providing the sequencing platforms for the researchers, mainly domestically within Japanese, And we're also doing some technological development by ourselves. And maybe the latter part of my talk will be somewhat associated with the long-read sequencing. So let me briefly introduce you some our recent publications for the long-read sequencing. For the short-read gene expression analysis, that will be the main body of my presentation. So, and for example, we have just reported the long-read cancer genome sequencing analysis, lung cancer genome sequencing analysis this time, and we show that the quite complicated pattern of the structural variations, are quite twisted pattern of the mutation can be detected. Firstly, by making use of the long-read sequencing analysis, which may not be possible without the help of long-read sequencing. That is almost impossible using the short-read sequencing. And we are doing the same thing, but not to the genomic sequencing, but also the full-length cDNA sequencing. And in this case, we showed that the aberrant splicing isoforms sometimes occasionally take place in cancers, and by coding a quite strange or abnormal, actually, the coding amino acid sequences, those isoforms can be served as an important source for the neoantigen, which are an important targets for the immune checkpoint therapy. So, and the other technological development, let me introduce our papers, is the long-read sequencing analysis of the methylation. So, in this case, instead of bisulfite sequencing, we made use of enzymatic mediated sequencing and conducted long-read subjected this template for the long-read sequencing so that we can analyze the methylation at the 5 kb in length, which is 10 times higher than the bisulfite sequencing. By making use of this approach, the phasing analysis of methylation is firstly enabled, showing that this chromosome is methylated or the other chromosome, for example, in case of the imprinting, genetic imprinting, we can separate which chromosome is methylated. And this is the usual phasing analysis of lung cancers. In this case, again, lung cancer genomes. And we can tell the difference between the chromosomal statuses of our sister chromatids, right? So some events are taking place specifically in either of the haplotype, the chromosomes. And we have two chromosomes, paternal origin and the maternal origins, and they are two independent molecules. So sometimes the different cancer mechanisms are ongoing depending on the chromosomes. So for further details, please take a look at the references and the review articles, which I have edited from Nature Spring Book and the special issue of the Journal of Human Genetics, which is also a journal of Springer Nature, and for several review papers. So I'm sorry for the lengthy introduction, and let me go back to the main story of the spatial transcriptome analysis. Our place is lucky, but I'm sorry, the map is written in Japanese, but this is our campus. The magnification is quite large because our campus is quite large. So the this length, this size of this size is about 2 kilometers. So which is a big campus. 1.5 exaggeration, sorry. So the lucky point of our campus is here. Next our campus is located just next to the National Cancer Center at East Hospital, which is part of the largest human cancer network nationwide. So they are having an operation, surgical operations of more than 1,000 cases per year for various cancer species, and almost always taking advantage of that regional advantage. Usually first target of our new platforms, new technologies are on cancer genome applications, because that's the kind of easiest specimens for our access. So let me introduce about our several stories about the Visium analysis, because I have not ever started to make use of your STOMICS approaches, and allow me to use the Visium, for example. And for the first topic, and we analyze some cases of breast cancers in this case, and we believe we have identified several of the very earliest events of the cancer development, from the relatively benign stage to the malignant stage. So, and for the remaining part, the second part, I would like to introduce some preliminary analysis for the integration of the long-read sequencing analysis using the Bayesian templates, which enable the full-length CNN sequencing of the Bayesian, which is also the main target of our collaboration possible collaboration with the BCI researchers making use of STLTR, which is a virtual long-read sequencing. And to be honest, the long-read sequencing, the physical long-read sequencing, in our case, we are using nanopore ProMethylene sequencing, but actually the throughput of this long-read sequencing is not as large as the short-read sequencing, even though a substantial progress has been made for their technologies. Anyway, so maybe I don't have to go into details about the importance of the spatial transcriptome analysis. We're firstly able to analyze intracellular heterogeneity and the microenvironment in cancer tissues. And without making use of spatial transcriptome analysis, those features are totally out of the access for the short-read sequencing or even single cell sequencing analysis. And in this case, we made use of Bayesian because this is the first round of the Bayesian platform, because our place was the first place where the Bayesian platform was introduced. I think it's the first in Asia Pacific, but at least in Japan. So we made use of the Bayesian spatial gene expression solution for the breast cancer specimens first. That was almost 3 years ago. 3 years ago. The paper was published last year. So, and the title was our efforts to elucidate the heterogeneous evolution of the ductal carcinoma in situ of the breast, in short, DCIS. And the DCIS, I don't want to go into the pathological details because we are not medical doctors. And this is the ductal carcinoma in situ. Just please remember that this is the very early stage, just the intermediate status from the benign precancerous lesions to the invasive cancers, which is an invasive ductal carcinoma, IDC in short. So the problem is a substantial number of DCIS cases are found in Japan and also worldwide. And some cases, they show never progress to the full grown-up form of IDC. So the first choice of the current treatment is the surgical operation. But somehow that the surgical operation seems to be quite over-treatment of this in some cases. But the safety for the sake of safety first reason, we had to make this operation. Actually, the cure rate is quite high, by the way. And so we want to know if we can separate the patients beforehand by molecular diagnosis, whether they are likely to develop to IDC or they will remain in a state of DCIS, the benefits from the patients will be quite substantial. And firstly, we conducted the usual clinical sequencing analysis. And in short, we identified two types of mutations, two groups of the patients having those mutations. One is the GATA3 mutation, and the patients having GATA3 is a gene, transcription factor, by the way. When the patients carry GATA3 mutation, the patients are likely to develop IDC, statistically relevant. And on the other hand, there are a group of patients having the PIK3CA mutation, another gene, which is this is a signal transducing molecule. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically significant sign of the better prognosis. And this is a kind of statistically sig However, we also notice that there are several substantial, I have to say, number of exceptions, right? For example, in some cases, GATA3 mutation did not always lead to the malignant transformation, but the vice versa. So we want to know why GATA3 mutation is associated with the worse prognosis and the PIK3CA for better prognosis on the molecular basis. So then we conducted the spatial sequencing analysis, Bayesian sequencing analysis. And this is the general sequencing analysis of the these days quite popular platform of the Bayesian analysis. And okay, this is the clustering analysis, and the tumors by the rough clustering analysis. This is the UMAP analysis. And for as far as the tumor plots are concerned, three clusters appeared separately depending on the status of the cancers. And for example, this is the pure DCIS in situ, remaining in situ within the milk duct. Okay, so the DCIS2 appeared. Another cluster appeared as in DCIS2, and which is a kind of invasive PERD, but not having experienced an EMT, epidermal mesenchymal transition. And IDC1, the blue dots, are the really malignant DCIS. So 3 parts. And I will not explain about details, but the stromal cells, normal cells, showed a different transcriptional programs, expression profiles, depending on the neighboring stage of the cancers, which will be the next target, actually, another topic of the talk. But okay. I will skip that part in my talk. So we especially focused on this part. And according to the clustering analysis, the inside, the green parts belong to DCIS part, the benign part, cluster 3. And the outside part, red dots, on the other hand, belong to malignant parts. So something should take place between blue and red dots. So we conducted a differential gene expression analysis by comparing these 2 parts, cluster 2 and cluster 1, the malignant and the benign parts. And as I forgot to mention that this is the pathological image. The inside is somewhat pathologically. Inside, the pathologist said that I'm not a professional pathologist. And according to the pathologist, inside part of the cells look like benign, having a smaller nuclear shape, and the other features show they are still a kind of benign. The outside are just becoming more malignant looking, enlarged nuclear and some irregular shape of the cell shape having become more malignant looking. So then the clustering analysis and the gene expression analysis followed. And we conducted the differential gene expression analysis like that between 2 clusters. And these parts can be done as a single cell analysis, even though each spot contains more than 10 to 20 cells. And actually, we could do the clustering analysis and we could do the differential gene expression analysis. And we found that the transcriptional module having the GATA3 transcription factor, GATA3 transcription factor as a core appeared as a responsible, maybe the core network, which substantially changed between the inside and outside part of the particular duct of the milk duct. So, and this this result appeared as a hypothesis-free manner, so which is consistent with the previous clinical sequencing analysis. So we further conducted some gene expression analysis for epidermal mesenchymal transition genes and angiogenesis related genes, and estrogen response-related genes, and found that the dimension fibronectin and clonine-free are included in the EMT-related genes, and many of those genes are upregulated just outside of the clusters where the malignant formation of the cells just took place. So our hypothesis is that the GATA3 triggered that change of the gene expressions. Therefore, the malignant transformation of the cells took place, and therefore, the worst prognosis of the patients was brought about. So, the important thing is that this GATA3 mutation is not did not take place at the genomic level. So we carefully looked at the genomic mutations at the corresponding regions and could not detect any genomic mutations. So in this case, transcriptomic changes are the main reason of the malignant transformation mediated by GATA3. And maybe I should have presented this slide earlier. This is an easier case maybe. And in this case, we conducted a similar analysis, and some parts appeared as the just place where the malignant transformation had just started. And again, we identified GATA3 transcription factor, the same transcription factor, as the transcriptionally key responsible molecule having the use of change for the EMT and the angiogenesis and other cancer-related genes in these regions, between these regions. And in this case, the GATA3 mutation was acquired when the cancer developed to DCIS to IDC. So the story may be simpler. In this case, the GATA3 aberration was mediated by the genomic mutation. So that is. In neither of the case, we found at least we analyzed 3 cases, and at least in two cases, GATA3 the aberrations either at the transcriptome level or genomic level appeared as a responsible factor. And on the other hand, we also conducted a similar analysis for PIK3CA side. And for the PIK3CA side, and obviously the malignant transformation was not observed, and the transcriptomic features are relatively monotonous, ubiquitous. There's several, not so many aberrations at the molecular levels were observed. So this seems to be totally kind of benign. So we're currently hypothesizing that the normal cancer developed to DCIS, and when something take place in the transcription network of GATA3, in some cases at least, and the cancer may develop start to develop to IDC via the GATA3 transcription network as a base to further accelerate the EMT or angiogenesis associated molecules, and which may not be the case in case the the PIK3C mutation was the first mutation where to transform the normal cell to the DCIs. So that's a kind of conclusion. Somewhat encouraged by the initial success, we are conducting the same analysis for a number of cancer specimens. And for example, lung adenocarcinoma cases, and making use of this for this analysis FFP specimens. And the big advantage of FFPE platform for the BESIM is that we are able to make use of the archival samples, either 5 years old or 2 years old, 10 years old. Specimens can be employed for the analysis, which is a big advantage. And another big advantage is the pathological view. The HE image was much clearer than fresh frozen samples. So we are making a shift to the FFP specimens, and several stories are developing. And I will not go into details, but for example, several cancer developing pathways are inferred from the analysis like this way. And maybe this cancer started from the well dissociated region here, and finally going to the invasive parts via the inflammatory and necrotic steps inside, in between. So by taking all the information together, maybe we have to deal with the mathematical modeling to finally explain the spatial the expansion pattern of the cancer development. But of course, obviously, the biggest problems toward that goal is the lack of the resolution. So there are several platforms announced to being developed for Visium, HD, and other platforms, but currently not yet. At least we don't have an direct access to 10x Xenium or your StereoSeq platforms. So at least for time being, we decide to make use of codex system, which is a multiplex immunofluorescent analysis, where the analysis can be done at the subcellular level. We can at least tell the difference between individual cells, each cell, by looking at the molecular markers, even though we can analyze, say, at least 50 proteins, genes at the same time. But the power is actually quite substantial. And when we're looking at the CD20 of the Visium, dots appear like this way. But when we also integrate the codex images, and much clearer or the more vivid images were obtained. So this case is CD20, and actually the inside of the residential macrophage is a big topic with regard to the association with the cancer cells inside. So when we look at the Visium, this is just no matter a dot, one single dot, but actually we are able to analyze the peripheral cancer cells in the context of this clearer image. So, and we are currently analyzing a number of the cancer specimens and integrating that image with this codex and trying to obtain further detailed pictures. So then another topic is that then the question arises, as far as time permits, I will try to explain this part. We come to want to see the genomic mutations. And so far we have been focusing on the transcriptome features of the spatial transcriptome because it's a transcriptome analysis. But the interpretation of the data will be quite different when some genomic mutations may have acquired in the middle. So, and let me skip these slides. And at least, and the idea is simple. We try to make use of the first amplicon templates of the Visium, right? Anyway, to do the Visium analysis, we had to firstly amplify the entire full-length cDNAs, and afterwards to deal with the short-read sequencing, we chopped that full-length cDNA into the 3' end pieces and do that first only for the sequencing analysis. So that's why the current Visium sequencing is on the 3' sequencing platform. But instead of making use of the short-read sequencing, we tried to use the full-length cDNA together with the spatial barcode for the long-read sequencing, in my case, Prometheum. So to make the long story short, after the enrichment was needed, actually, because the gene expression levels of the driver genes is not always low, not always high. So we had to enrich that gene expression levels, and by making use of the age-length hybridization capture with some modifications, because we have to combine, made a modification to enable the current short selector is designed for the short-read sequencing. So if we apply the same protocol for the long-read sequencing, the data will be quite messy, having a lot of the non-target region captured. But we were able to modify that part, and currently we can enrich the long-read full-length cDNAs at the ratio of 2 to the 5.5, which is 50 times higher than the non-captured sequencing. So by looking at the captured enriched long-read full-length cDNAs, we were able to identify several driver genes, transcripts of the driver genes, together with the spatial barcodes. I forgot to mention, but the Q20 introduction of the Q20 is another key. For the promethyl sequencing, the quality of the sequencing is greatly improved to more than 99%. So which is quite an important factor. But anyway, and this place is the ACFR exon 19 deletion lesion, and this is the poly A part, and this is the spatial barcode, UMI part, and the spatial barcode part is this way. And by looking at this sequencing, we were able to pinpoint this sequence tag was from this spot, and so is the case in another case. And so as a total, we're further polishing the bioinformatic pipeline. At this, we were able to identify 44 spots having DAS mutation in this, starting from the DISM template. The spatial mapping of the transcript, the genomic mutations was enabled by this way. And then, sorry for the KRAS mutation in other specimens. So I'm sorry, the time is already up. To summarize, we have studied the spatial sequencing of the breast cancers at first using the first generation DCA platform, and then we are moving to the large scale, the scaling data analysis for various cancer specimens. This time in using FFPE, and to further improve the detection, to further detect the genomic mutations in the DSM planar, we are trying to develop a method enabling that full-length cDNA sequencing. So the last slide is the arrangements, and I think my time is already up, and I would like to thank all the listeners for your kind attention. Thank you.